The overall goal of this procedure is to quantify behavioral and cognitive changes induced by cranial irradiation. The first step is to test hippocampal-based spatial recognition memory using the novel place recognition task. Next, anxiety levels are assessed using the elevated plus maze task. Fear-based memory formation using the hippocampus and amygdala-dependent contextual fear conditioning task and the amygdala-dependent cued fear conditioning task is then measured. The final step is to administer the Morris water maze task in order to assess spatial learning, spatial memory, and reverse learning ability. Ultimately, this protocol detects changes in mood, spatial and non-spatial learning and memory, as well as any sensory motor disruptions induced by cranial irradiation.
Though this method can provide insight into the effects of cranial irradiation on behavior and cognition, it can also be used to other situations in which cognition is impaired as an unwanted side effect of other therapy, such as in the case of chemobrain or chemotherapy-induced learning and memory impairments.
Individuals new to this method will struggle because of the time-consuming nature of setting up and optimizing the equipment. Interpretation of the findings can be difficult if experimental procedures are not tightly controlled, and variation in the data makes it difficult to detect differences between control and experimental groups.
Visual demonstration of these methodologies is critical so the reader can appreciate the subtleties involved in organization of the experimental rooms in which the cognitive testing is conducted, as well as the specific procedures employed in order to ensure consistent handling of the animals.